Good afternoon and welcome to the 2006 Alice Louise Reynolds Women in Scholarship Lecture. I am Brian Champion, the coordinator of library lectures and the political science librarian, and it is our distinct pleasure to have with us today Professor Laura Bridgewater from the Department of Microbiology and Molecular Biology, who will deliver an address entitled BMP-2, an old protein in a new place. We will hear more about Professor Bridgewater in a moment, but first, the Alice Louise Reynolds Women in Scholarship Lecture began as a tribute to one of the great women scholars of BYU, and to someone who was a great benefactrix to the BYU Library. Over a long association with BYU, Professor Reynolds taught comparative literature and English to literally hundreds, if not thousands, of eager BYU undergraduates. Through her example of devotion to words and stories and to the restored gospel, Professor Reynolds' faithfulness and intellectual excellence inspired generations of BYU students. In her later years, students who had departed BYU still remembered her influence on their quest of lifelong learning and organized Alice Louise Reynolds clubs so that they could socialize and continue their love of literature and collegial discussion. Her personal library was donated to the fledgling BYU Library nearly 50 years ago, and this gift constituted the core of the library collections. In her honor, and to recognize the academic and spiritual contributions of women faculty at BYU, the library began this tribute lecture to Professor Reynolds to memorialize her significant contribution to BYU. The library is pleased to host this lecture as recognition of BYU's female scholars. Following Professor Bridgewater's address, we will be pleased to have a brief question and answer session. Now to today's featured speaker. Professor Laura Bridgewater from the Department of Microbiology and Molecular Biology is one of us. She received a BS degree from BYU in 1989 and earned a PhD from the George Washington University in 1995 and returned to BYU in 1999. Along the way, she has completed extensive research into the effects of chromium-mediated DNA, and her research, with her as principal investigator, has been published in such prestigious scientific journals as the Journal of Biological Chemistry, Molecular Carcinogenesis, and Toxicological Sciences. In addition to her research and publishing, Professor Bridgewater is a mentor to undergraduate microbiologists, and she is generous with her time for student consultations. A gifted teacher, she is mentor in the spirit and mold of Professor Reynolds and embodies the desirable attributes of faithful scholarship for which Alice Louise Reynolds is most famous. It is with a great deal of appreciation the library is pleased to introduce today the 2006 Alice Louise Reynolds Women in Scholarship Lecturer, Professor Laura Bridgewater.
It's a great honor to have been selected to present the Alice Louise Reynolds Women in Scholarship Lecture today. I've enjoyed learning about Alice Louise Reynolds as I prepared for this lecture, and I'm honored to have my name associated with hers in a small way. She made wonderful contributions to the university in its early days, and we all continue to benefit from her vision and her determination in establishing a world-class library at Brigham Young University. Furthermore, just by being here and doing what she did, Alice Louise Reynolds paved the way for women at BYU, both as faculty and as students. Her efforts helped shape a university where the contributions of women are welcomed and appreciated. I would like members of the Harold B. Lee Library faculty and staff to know how much I appreciate being selected for this honor. Before I begin talking about my research, I want to acknowledge some very special people here in the audience today. My dear husband, Tim, who married me before we ever knew I would become a scientist. My 4 children, Chelsea and Nate and Brandon and Lily, who were all born while I was in graduate school and have never known anything but a scientist mom. And my parents, Lawrence and Carol Clark, who raised me to believe I could do anything I wanted to do with my life. I'm so proud to have you all here today and excited for the chance to explain a little bit about what I spend my time doing when I'm at work. Now, the story I want to tell you today is about a protein called BMP-2, which has been studied for the past 20 years and was thought to be pretty well understood. This story illustrates something I love about science. It will never be done. There will always be new and exciting things to discover, and just when we think we have it all figured out, we're most likely to stumble on something entirely unexpected. What could be more fun than that? We need to begin today with some basic molecular biology background. You've all heard of genes. Genes are made of DNA. Genes carry all the information a cell needs to make every protein that it has to make. The proteins then do pretty much everything that a cell needs to have done to stay alive. When a cell divides, it first has to make a copy of all its genes so that both daughter cells have a complete set. As long as they each have all the information, they can both go on to make all the proteins that they need to live and to grow. So how is a protein made? The DNA is housed in the nucleus of a cell, right here. The DNA sends out a message with directions on how to make a particular protein. This message leaves the nucleus and it carries that message. It enters the cytoplasm, which is the rest of the cell outside the nucleus. And once they're in the cytoplasm, its directions are read and followed to build the correct protein. A cell usually has thousands of different messages leaving the nucleus and thousands of different proteins being built from those messages at any given time. So once built, what do these proteins do? Like I said before, they do almost everything that has to be done in a cell. Some proteins go to the nucleus. So they're made out here, they go back into the nucleus, where they might do things like helping to copy the DNA, or turning on other genes so that they can make their messages and send those messages out with directions for building other proteins. Some proteins participate in forming the structural components of the cell, these other things you see in the drawing. Other proteins are secreted from the cell. So they leave the cell entirely. They can move through the space between cells to bind the outside surface of other cells and send a signal, allowing communication between cells. The protein that I want to talk about today has been known for the past 20 years as one of the kind that's secreted from cells. BMP-2 stands for bone morphogenetic protein 2. And when BMP-2 is secreted from cells and binds to the outside of other cells, it signals those other cells to start developing into bone cells. So that means, of course, that BMP-2 is very, very important for skeletal development. But BMP-2 is even more interesting than that because it's also involved in other developmental processes besides bone formation. BMP-2 plays a role in heart development. It promotes the differentiation of neurons, that's nerve cells. It provides positional information in developing limb so that those cells know if they should become a thumb side digit or a pinky side digit. And it also seems to play a role in helping cells know when it's time for them to die. Now, to me, it seems really surprising that this one protein can have so many very different functions. Now, for the last few years in my lab, our research is focused on studying the kind of proteins that go back to the nucleus, back into here, and activate other genes, which then send out their messages to make other proteins. We work a lot with nuclear extracts, which means we collect just the nuclei of the cells, and then we break them open and we extract the proteins inside the nucleus to study. And a couple of years ago, we were using two different techniques to identify proteins in the nuclear extract, and both techniques identified BMP-2 there. Now, the first time it happened, we dismissed it as contamination because everyone knows that BMP-2 is a secreted protein. It leaves the cell, it doesn't go back to the nucleus. But when it happened again, we decided to take a closer look at BMP-2. And what we found has opened a whole new avenue of research in my lab. This picture illustrates what was already known about the structure of BMP-2 and how it's made. A protein is a chain of amino acids. Think of an amino acid as a railroad car, and the protein then is a whole freight train. The protein has to fold into a complex three-dimensional structure before it becomes functional, but to make it easier to visualize, we often represent proteins as straight lines or bars, like this. When proteins are synthesized, the amino acids are chained together one by one, from this end, single amino acid at a time, to the other end until the complete chain is finished. To make BMP-2, the message or the instructions for building BMP-2 are made in the nucleus, like all those messages, and sent out to the cytoplasm. there translation occurs. Translation is what we call the process of reading the message and chaining the amino acids together in the correct order specified by the message. The first 20 or so amino acids that get chained together in BMP-2 constitute a signal peptide. That means they signal to the cell that the protein that's in the process of being made needs to go into the pathway that leads to secretion. This pathway begins in the endoplasmic reticulum, which is this structure over here. So the message leaves the nucleus. The first few amino acids are chained together, or translated, and that tells the cell, take this here. We call that, by the way, ER for short, endoplasmic reticulum, ER. Makes it much easier to say. So once there, that protein is translated in such a way that the protein ends up inside the ER when it's finished, inside these little pockets, and gradually makes its way through from one to the next until it reaches the outside of the cell and can be secreted. Once the signal peptide has succeeded in directing the protein to the ER, the signal peptide is cut off right there and discarded. Has no other purpose. Once synthesis Once synthesis is completed inside the ER, the protein undergoes a second processing step in which it's cleaved at another site. The end of the BMP-2 protein is released from the rest of the protein chain, and it then pairs off with another identical cut BMP-2 protein end, just like this, and those two together constitute the mature BMP-2 molecule that's secreted from the cell and moves away through the space between cells to signal other cells. The part that remains we call the propeptide, and it has previously been assumed to just float away and be degraded. When our experiments suggested that BMP-2 was present in the nucleus of the cell, even though everyone knows it's a secreted protein, we decided to examine the amino acid structure of BMP-2 to see if it contains what we call a nuclear localization signal. This is another one we abbreviate, NLS for short, nuclear localization signal. And NLS is a particular amino acid sequence found within a larger protein that causes that protein to be delivered to the nucleus, much the same way that the signal peptide causes a protein to be delivered to the ER. So we used a computer program that can predict possible NLSs, and it analyzed BMP-2 and predicted 3 of them. The first 2 predicted NLSs were here, the one marked A, and there, the one marked B. The third one was located here. You can see it overlaps that B site, and it also overlaps the site where the protein has to be cut in order for mature BMP-2 to form. We expected that this one would probably not be functional because of its location there overlapping that cut site. But we were pretty excited about these other 2. They bracket the part of the protein that's left when mature BMP-2 is cut away. So we thought maybe this propeptide, rather than just being degraded, maybe it might go to the nucleus and have some important independent function there. Now the computer predicts these NLSs based on similarities with other known NLSs, nuclear localization signals from other proteins. That doesn't mean they will really be functional. You have to test them in cells to know for sure. So to test the predicted NLSs, we constructed artificial genes that made proteins containing each predicted NLS, like that one marked A, fused to a green fluorescent protein. That's what GFP stands for, green fluorescent protein. Now, if the NLS is really functional, it should carry the whole protein to the nucleus of the cell, and we'll be able to tell it's there because we can see the green fluorescence. If it's not functional, if the NLS doesn't work, the GFP will be all over the cell. We then grew cells in culture, and we put these artificial genes into their nuclei and watched to see what would happen. Here's what we saw when we tested the first predicted NLS. Green, of course, is the green fluorescent protein. The red that you see in these pictures is a special stain that stains just the nucleus. So that lets us see where the nucleus is so we know if the green fluorescent protein went there. And you can see that the first predicted NLS was completely non-functional. The GFP is everywhere throughout the cell. Here's the nucleus, there's the green fluorescent protein, here are the two images superimposed, green is everywhere. So we then tried the second one, the one we named B, and we got the same result. There's the nuclei, green fluorescence is all throughout the cell. Next, we tried this arrangement because remember, in the actual BMP-2 protein, these two predicted NLSs bracket that propeptide. So we thought maybe that's what it takes. You have to have both of them together to carry the protein to the nucleus. That didn't work either. As you can see, there's the nucleus, two nuclei, there's two cells. There's the green, and it's throughout the cells, inside and outside the nucleus. Last of all, we tried that third NLS, this one that overlaps the site of cleavage. This one, to our surprise, showed clear nuclear localization. There's three nuclei have three cells, and you can see the green fluorescence is confined to those three nuclei. There are the images superimposed. So, that tells us that this nuclear localization signal is functional, except that if it gets cut in half, it would be destroyed. So what that means is that the version of BMP-2 that goes to the nucleus must be an uncleaved protein with the NLS still intact. How can that be? Because everybody knows BMP-2 is cut right there. One possibility we thought of is that the cell might have a way to prevent cutting when it needs the nuclear version of BMP-2. And then that uncleaved BMP-2 would have to somehow work its way back out of the ER and go to the nucleus. To test this, we used a compound that you can put on cells and it goes into the ER and prevents cutting. We figured that this should result in much more nuclear localization of the BMP-2, but we were a bit disappointed to find that inhibition of cutting didn't really affect the amount of BMP-2 that went to the nucleus. So then we started thinking, what if a version of BMP-2 was made that didn't have the signal peptide at the front end, was missing that? Then that protein would not be directed to the ER for translation, which means it would never encounter the cutting machinery or be secreted. Instead, it would be translated in the cytoplasm and the NLS would stay intact. So, how could you possibly get a version of BMP-2 without the signal peptide? Well, it turns out that some messages for making proteins contain ambiguous information about where the protein synthesis should begin. And we wondered if maybe the BMP-2 message was like that, and there might be an alternative place the protein synthesis could start that would leave out the signal peptide. Using computer analysis to examine the gene, we did find a possible alternative site right there. Use of this start site would produce a version of BMP-2 like that, which would never enter the ER because it lacks the signal peptide. Instead, it would be translated in the cytoplasm. Now on this picture, these dots out here represent proteins that are being translated in the cytoplasm as opposed to the dots lining the ER, which are depositing proteins into the ER. So if this protein ended up being made in the cytoplasm and the protein never encountered the cutting machinery, the NLS would stay intact. The whole protein then could be delivered to the nucleus. Seems reasonable, but how can we know if this shorter protein is ever actually made? We made a special version of BMP-2 that can make BMP-2 protein in a test tube. And we can do that without ever having to put the gene into cells. And we did that, and then we looked at whether there were other shorter proteins made in addition to the full-length protein that corresponds to the predicted size. Let me back up. We were looking for a protein that might be this size rather than that full-length size. So let me explain what you're looking at here. When we make proteins in a test tube, we can't just look at the test tube to see what proteins we've made because they're too small. So what we have to do instead is we make those proteins radioactive and we separate them on a gel. We load proteins at the top of the gel. What you're seeing here is a section of a gel. So we load proteins at the top and we apply electricity to pull the proteins through the gel. Small proteins go through faster than large proteins, and so we end up with proteins separated on the basis of size. To see the radioactive proteins, though, we still can't just look at them and see them. We have to place X-ray film against the gel. And then we see dark bands showing up on the X-ray film where the proteins were. This lane right here contains protein made from the normal BMP-2 gene. And this band right here, the darkest band, contains the full-length BMP-2 protein of the size that we expected it to be before cutting. Now you can see here that we do have some lower bands. We know that there are some smaller proteins made, but we can't tell just by looking exactly where translation started to make these lower bands. In fact, we can't even tell for sure that they are BMP-2. They could be some other contaminant in the system. So to find out for sure if either of these bands contain protein, BMP-2 protein, made by beginning translation at the alternative start site, we mutated the possible alternative start site. So we took this version of the gene and we changed the site right here so that it could never possibly serve as a start site. And we then made that protein in a test tube. And that's what you see in this lane. We found that when we mutated that possible alternative start site, this protein band didn't show up. So that tells us that the band did in fact contain a protein that was made by starting translation at that site, right there. Furthermore, we know that the BMP-2 protein made by starting translation there at that second site would have no signal peptide, it would not be translated in the ER, and so it would avoid being cleaved and secreted. Instead, the NLS would stay intact, and that protein could go to the nucleus. Now that we knew what we were looking for, we were able to go back to nuclear extracts from real cells, not in the test tube anymore, but from real cells, and find that the BMP-2 in the nucleus is, in fact, the size that we would expect if translation started at the second site. In addition, we put these genes into real cells to make sure that the same thing would happen in real cells as we saw happening in the test tube. And we found that when we put in the normal gene that would make this protein, we saw most of the time we would see BMP doing what everyone has seen it do for years. 20% of the time we would see BMP-2 in the nucleus. When we mutated the first start site, before we mutated this one to see how that would change things, we made a version where we mutated this, which meant translation could only start at the second site to make this protein. 90% of our cells had nuclear BMP-2. So that was pretty exciting. From all these experiments, we know that translation of the BMP-2 message can begin at this second start site, and we know that when it does, we get BMP-2 in the nucleus. So after all these experiments with engineered genes in test tubes and in cultured cells, we went back and looked at a couple of additional cell lines that had not been manipulated in any way. Now this is different from the slides I've shown you before. This green is not green fluorescent protein. These cells were just grown up on a slide and they were stained with something that shows up green wherever the cell's own naturally occurring BMP-2 is located. And again, the red stain shows us where the nuclei are. We were able to find both fibroblast and mesenchymal cells. 10T1/2 is a fibroblast cell line, BALB3T3 is a mesenchymal cell line, and in both of those cell lines, we were able to find cells that had naturally occurring BMP-2 in at least some of their nuclei. So having discovered this well-known protein in an entirely unexpected place, the obvious question is, what is it doing there? Answering this question is the focus of a major research effort going on in my lab right now. We suspected right away that the nuclear version of BMP-2 might be involved in some of the other functions besides bone formation that I told you about, perhaps heart development or nerve cell differentiation. To see if our suspicions are correct, we are using two different experimental approaches to answer this question of what BMP-2 does in the nucleus. One approach is being spearheaded by Jamie Mayo, a PhD student in my lab. She's constructing what we call a knockout mouse. A knockout mouse is a mouse that it's been engineered to contain a single mutation inactivating or knocking out a single gene. The mutation's introduced at the earliest stages of embryonic development, so that we can then examine the developing mouse and we can see what goes wrong when the gene of interest isn't working. And by seeing which tissue or structure or organ system doesn't develop properly, we can learn something about the normal role of the knocked out gene. Jamie has faced an enormous challenge in this endeavor. She cannot just knock out the entire gene as people usually do, because that would eliminate both the secreted and the nuclear versions of BMP-2. And we already know that the secreted form is essential for normal embryonic development. So knocking out that secreted form won't convince anybody that the nuclear form is important for anything. So instead, Jamie's had to devise a way to knock out only the nuclear version of BMP-2 while still allowing the secreted BMP-2 to be made normally. Only in that way can we study the function of nuclear BMP-2 without being confused by the secreted form. This has required some very sophisticated genetic engineering, and I can't begin to explain it all in the time that we have here today, but this flowchart illustrates the many recombinant DNA steps required to complete this construction and to introduce the desired changes into the mouse DNA. Now, Jamie has already accomplished most of these steps, and she's now about to begin generating the mice. By examining the mice that result from her work when the secreted BMP-2 is present, and it functions normally, but the nuclear BMP-2 is not made at all, we'll be able to learn whether the nuclear version of BMP-2 is required for correct development of the heart, the limbs, the nervous system, or anything else. Second approach that we're using to figure out what BMP-2 does in the nucleus is focused on identifying other genes whose activity levels may be altered by nuclear BMP-2. If we can identify those genes and we know something about what they do, that'll tell us something about what nuclear BMP-2 does. So to learn how nuclear BMP-2 may influence the activity of other genes, we've used a fairly new technology called microarray analysis. It allows us to compare activity levels of the same gene in two different samples by comparing the amount of message that the gene is producing. And the results from such an experiment look something like this. The upper image you see is a close-up of one of these squares down here, which represents a larger piece of a microarray. The spots up here, each spot represents an individual gene, a single gene. If the spot's dark, like that, that tells us that neither of the two samples we're comparing is making much of this message. If the spot's green, it means that one sample is making more of it. If a spot is red, it means the other sample is making more of it. And if the spot's yellow, like you see some of these here, that means both samples are making lots of that message, and that's not too interesting to us. We're not interested if neither sample makes it or if both samples make a lot. We're interested in the differences. For our experiments, we compared normal cells with cells that had been engineered to produce large amounts of BMP-2 in the nucleus. We wanted to see, when you have lots of BMP-2 in the nucleus, what other genes change their activity levels in response. We found over 300 different genes whose activity levels were increased or decreased in response to the presence of BMP-2 in the nucleus. Our challenge now, though, is to sort through all that data, confirm its biological relevance, and figure out what cellular processes those genes contribute to. As I said, this work is still ongoing, and we have yet to submit it for publication, but we do have some very tantalizing preliminary evidence that when BMP-2 is in the nucleus, it changes the activity levels of genes involved in both nervous system development and cardiovascular system development. So we are very excited about these preliminary results and working very hard to confirm them. So we have this protein, BMP-2, which is known to be involved in an extraordinarily broad range of different processes. The discovery that there are actually two different variants of the protein that act in entirely different cellular locations and by entirely different mechanisms helps to explain how this is possible for one gene to have so many different functions. Isn't biology amazing? I love it. I just can't imagine anything more interesting than the study of life. On the one hand, it's so complex. And yet, there's such a beautiful simplicity about the molecules of life. DNA looks the same whether it comes from a human or from a mouse or from a fish or from a plant. It's all DNA. And to me, the goal of biological research is ultimately to figure out how God did it. Sounds overly ambitious, I know, and we're not likely to succeed or achieve it in this life. But to me, that's what it's about. Try to understand how it all works. And every little discovery is such a thrill, and it reveals one more beautiful little piece to the story. And better yet, we've been admonished in Scripture to study biology. Did you know that? Really? We're told in Doctrine and Covenants, section 88, says, teach and study about things both in heaven and in the earth and under the earth. That's biology. Of course, we've also been told to study and learn about lots of other things, but no one person can study all those things, and I'm just as happy as I can be that I get to study biology while other people work on things like the perplexities of nations. Well, I've enjoyed this chance to share my research with you, and in closing, I need to give credit where credit is due. The work I've described here has been funded by the National Institute of Arthritis and Musculoskeletal and Skin Diseases, which is part of the National Institutes of Health. The work I've presented was mostly performed by Jenny Feline and Jamie Mayo. Jenny Feline was a research associate in my lab for about 3 years. She recently left the lab and returned to Sweden, where she's originally from. We miss her very much. And she did lots of the work that I presented and had begun the microarray analysis, which now Jamie Mayo has picked up along with the knockout mouse that she's been working on. Jamie Mayo also has been working with a brand new student in the lab, Sam Wilkinson. He's our newest undergrad and is working to help confirm the microarray data. We also have lots of other students in the lab who aren't working directly on this project, but who contribute so much just by being there and working on their different projects. I have also worked quite closely with and had significant help from Dr. Chin-Yo Lin of the BYU Department of Microbiology and Molecular Biology. He's worked to help us analyze our microarray analysis, or our microarray data. And then we've also had a lot of help and worked closely with Dr. Jeff Barrow of the BYU Department of Physiology and Developmental Biology on our knockout mouse construction. And these have been wonderful people to work with, and I've learned so much from them. I appreciate your attendance here today and your support of me and of the Alice Louise Reynolds Women in Scholarship Lecture. Thank you for being here.